Okay, and yours out there, hold on, sorry.
You have to download this.
Okay.
I will edit out.
All right, Arthur Porter, Florida Museum, thank you.
All right, let's see. Very good. Thank you all very much. Thanks IDIC Bio folks for the invitation to take part on this. So today I'm going to talk what's a very, and I can't emphasize this enough, a very recent interest of mine. And I wanted to talk a little bit about some work I'm doing with regards to color. But why color? As you all know, and I don't have to spend too much time on this, color is a critical component of organisms, right? Like it plays a wide variety of functional roles within species, both in terms of signaling, camouflage, mimicry, and even temperature regulation. But despite this being such a critical component of organism morphology, color is something for which we have very little consensus on how to measure and compare across species. So especially when you compare with size and shape traits, where we have 3D geometric morphometrics as a tool set that anyone can use in any context, the tools that we have for color approach the problem in very different ways, and there's very little consensus on how we actually should do that. And even more important than that, they're almost always restricted to the 2D realm. So we really don't have the ability to process 3D color information or a good tool set to process that information effectively. So some of this is natural, right? We know that dealing with color can be a lot more complex than doing just size and shape, in part because there's a lot of sources of variation. So there's sources from biology, like if you have mussel shells and they all have different colors, but they also have different shapes. So how do you do the mapping from this shell to this shell is not a trivial aspect. The other thing is that most images that we capture are to a large extent standardized. So you get situations like this where you have two pictures of the same specimen, one under diffuse light and under direct light. And as you can see, they look remarkably different in terms of their RGB values. And finally, there are things like UV, right? So when you take an image of these two species of the same genus of butterfly, under traditional light, they look fairly similar in their coloration patterns, but then you throw them under the UV light, the second half, and they look completely different. So it's natural that color is harder to deal with, and there is less standardization of how we approach this. But even then, I was surprised when a few weeks back, Zach and Laura arrived in my office and they asked me like a simple question. They were like, look, we have this photogrammetry model of sclera ossicle from Mahi Mahi, and they wanted to do a simple quantification, right? They wanted to know what percentage of the area was bone, what percentage was cartilage. And my intuition, when they first approached me, was like, oh, okay, this is going to be simple. That would be something that we can use to just, you throw that into a software and it's going to spit out like the area that has kind of reddish wine color and the area that is blue. But we were surprised to find they actually don't have those tools. Those tools simply don't exist. So spent some time and created a slicer module that basically does that. So it's tentatively titled Biomodel Colors. And the module basically requires two inputs, right? You need a 3D model of your object and the texture file, and then it has a very simple pipeline. So it starts essentially by wrapping the texture around the 3D model. So you have the texture in 2D and the 3D model wraps it around. So that, and it actually maps the color information through each vertex of the mesh. So this is each point in the mesh has an RGB value associated with it. From then, you can perform any kind of clustering method. We opted for k-means clustering with 2 clusters, right? Because in this case, we wanted to separate that into a binary model where it was either bone or cartilage. So you got this binary model in which every vertex color gets replaced by the centroid of the cluster. And once you have that, you can just split your mesh into 2 pieces and segment your 3D model and calculate some kind of measurement of what percentage of that mesh is bone, what percentage of the mesh is cartilage. So that worked well for the kind of thing that they were aiming for. But ultimately, it's something that is like not that satisfying, right? Because we work in biology, and biology is comparative. So how do you go from an approach that works well for a single structure and that does the job that you aim to one that works with another one of Zach's amazing data sets with the freshwater mussels? So how do you go from one to the other? So today I just wanted to briefly present a proposal for myself of how we might approach that problem. So ultimately, I think we will only be able to deal with color information and find a tool set that can deal with that information in 3D if we incorporate that color, our color tools to our 3D morphometric tools. And essentially because morphometric tools are basically tools that aim at doing one thing, which is to place specimens in a shared coordinate system. That's what 3D morphometrics does. It places individuals in a population in the same coordinate system so that we can quantify and compare these things across individuals. So in my view, we're only going to be able to deal with color effectively and create tools that quantify and compare color effectively if we place those individuals in a shared coordinate system in which you can say this point in this individual is equivalent to this point in this individual, and you can therefore use traditional quantification, principal component analysis, whatever you want to do with that kind of data set. And really those tools exist. Pieces of those tools exist. So just an example pipeline that one could use that I just applied to the freshwater mussels that Zach has collected. You can start with your samples. In this case, the 3D models are samples. You can collect semi-landmarks in these in kind of a grid fashion, and they don't even need to be in homologous positions or anything. So then you can use landmark transfer tools such as Alpaca, where you use deformable registration to map one 3D model into another. And then you can get these sparse homologies, so to speak, into dense homologies, where with tools like DECA by Sarah Roth, where you then basically establish point-to-point homology at every point in every 3D model to every point in another 3D model. So essentially, each individual in the population then becomes a deformation of a mean mesh and associated statistical shape model that describes the variation in the population of that mean mesh. So essentially, each individual in the population then becomes just a deformation of that mean shape. And with that, you can then deform all specimens that you started with into the mean shape of the population, and therefore created this shared coordinate system in which color information is mapped into. All right? With that, then you can then go and use more traditional color tools and then perform, for example, you can perform an initial color clustering, so you reduce the amount of noise that is associated with lighting and all that kind of sources of variation in color information. Obtain color maps for individuals. You can reduce those color maps using clustering techniques so that you merge colors that are too similar. And then you can merge those color maps together again by placing all color maps of the different individuals in the population, doing hierarchical clustering of those colors. Again, creating a threshold where you merge colors together. And in the end, you can impose this shared color space at every single individual in the population. So in the end, you end up with individuals that are not only in the same coordinate space in terms of spatial traits, but also in the same space of colors, so to speak. And when you're at that situation, then you can just, it's data that can be just played with. And you can do things like, for example, do a principal component analysis of the color information across individuals, find clusterings in that space, even among individuals that if you look back at the slides, they're completely different in shape. But when you actually place them in these shared coordinate systems, you see that their colors are all, they map to the same region of that space. And you can calculate similarity between individuals, find individuals like this one. In this case, this is a 2D plot, but if you actually had more dimension of this plot, this individual would fall completely outside of the rest, and which is this individual here, which is not similar to anything, and it lies at its own by itself. And with that, we finally perhaps can have a quantitative framework where we can analyze color in the same way that we analyze size and shape traits. And that's what I had for today, and I want to thank a bunch of folks that helped in the process.
So we have, it's time for question and answer again. Austin Mavs